Hello, my name is Kira Nomura. For my Biology 7 oral presentation, I chose to focus on Alaskan brown bears and foraging, specifically on stockeye salmon in southwestern Alaska. Before delving into the details of the study, we will briefly look at the impact and importance of Alaskan brown bears, which specific name is Ursus actos, foraging on Pacific salmon, Oncorhynchus and Oncorhynchus nercta for stockeye salmon. The relationship between brown bear and salmon is heavily influential on the population dynamic of either species, namely the brown bear, as it serves as a pivotal food source annually during the summer season. Aside from the impact that the relationship has on the species involved, there is an important secondary role that the brown bear's foraging on salmon plays. It provides an influx of nutrients and energy to the freshwater and riparian ecosystems in Alaska. This act of foraging was previously found in another study to have greatly enhanced the nitrogen presence in the forest as compared to areas inhabited by either species independently of one another. The spread of nitrogen and nutrients is accomplished by the brown bears dragging salmon carcasses and leaving behind unconsumed salmon flesh to deposit these nutrients. Additionally, this impact is further spread through excreted nutrients after the bear has consumed and digested the salmon. Understanding the pattern of foraging at ecologically relevant habitats, in this case spawning streams, will pave the way for biologists to study the process by which salmon nutrients and biomass are transported between terrestrial and aquatic environments. The study I chose to focus on is titled Alaskan Brown Bears Aggregate and Display Fidelity to Foraging Neighborhoods While Preying on Pacific Salmon Along Small Streams. The study was conducted at 6 stream sites surrounding Lake Aleknagik of the Wood River system. The lake is shown in gray, and the 6 streams that will serve as study sites are Happy, Hansen, Eagle, Bear, Yako, and Whitefish. For this study's purposes, the 3 streams on the northern side of the lake will be a trio, and the 3 streams on the southern side will be another. Each trio will also be referred to as a neighborhood in later slides. In order to access and keep track of brown bear abundance and movement at each stream location, non-invasive genetic sampling is employed to distinguish between individuals. The samples will be collected using unbaited barbed wire hair snares. The location of the wires are indicated by the red circles on the map. The study was conducted over 4 years from 2012 to 2015 for 6 weeks per summer. Prior to the study, it was predicted that a larger number of brown bears would opt to forage within one of the 2 trios. This is likely because there is a high cost of energy needed to traverse the lake or to travel around it. Additionally, bears would be more likely to remain loyal to the same locations as in the year or years prior, with the thought that the familiarity, salmon population dynamic, and stream characteristics would attract revisitation. Lastly, it was predicted that a larger female abundance would be recorded due to past studies showing that females are less sensitive to human presence than males. Although the study aimed to have a non-invasive approach, the brown bears still have some level of human exposure due to hunters and fishers that still work in the area. The study sites were carefully selected such that the 6 streams chosen were similar in size, less than or equal to 6.4 kilometers long, with an average of 2 kilometers separating the streams within the same neighborhood. In all 6 streams, salmon spawned in the lower 2 to 3 kilometers of the stream, and all spawned predominantly stock-eye salmon, with other species of Pacific salmon being rarely seen. This was selected to minimize any outlying factors that could cause any discrepancies in the results. Lastly, the study site should have minimal human presence, but since hunting and fishing, as previously mentioned, are still permitted in some areas, 100% human absence could not be attained. To collect data with a non-invasive genetic approach, researchers used 2 strands of 4-prong, unbaited barbed wire that were deployed at each location. Each strand was 8 meters long, with 12 centimeters separating the barbs along the wire. The wire was strung directly across the stream and hung to trees at locations past the first 2 kilometers of the stream. The wire was moved periodically to prevent habitualization. The hair samples were collected every other day using sterilized forceps to prevent sample degradation. After collection, the barbs were also sterilized by using a butane torch to burn them. The capture mark recapture, or CMR system, was used to estimate the number of male and female individuals. Genotyping was accomplished through PCA analysis in Genoplex and generated for a sample using 10 nuclear DNA, microsatellite loci, and 1 sex marker. Each sample was then amplified 2 to 4 times through a polymerase chain reaction, or PCR, to increase the accuracy. A successful match in genotype is either identical to one another or has one allele mismatch at 2 or fewer loci. The probability for an unrelated match or a match between siblings is very low. A population estimate was not determined due to the limited sampling period. The result of the genetic analysis yielded that 2,026 samples were collected. The samples were further subsampled in order to remain cost efficient. Of the 2,026 that were collected, 829 were submitted for genetic analysis. 524 of those were successfully amplified. Over the 4 years, the study detected 121 unique individuals, 68 females and 51 males. The amount of single detections, meaning that the individual sample was only detected once within a given year, varied across the years, 56.25% in 2012 to 36.51% in 2015. Annual numbers of detection per individual is 3.83 plus or minus 4.47 standard deviations for females, and 2.50 plus or minus 2.76 standard deviation for males. To the left is a chart that depicts the abundance estimation between male and female brown bears at the 6 locations. The dark gray shows female abundance, while the light gray is for male abundance. The black line indicates the 95% confidence interval. This figure is only focused on data collected from 2013 to 2015. The 9 individuals who were only detected in 2012 were excluded. In all years, at Happy, Hansen, Bear, and Yako, female abundance is higher than male abundance. Male abundance is higher 2 out of the 3 years at Eagle. In all comparisons, the overlapping 95% confidence interval indicated that the differences in male and female abundance at a given lake per year is not statistically different. The result of the neighborhood stream use from 2013 to 2015 showed that individuals detected at more than one stream in a neighborhood in a given year was a significant portion of the sample population. Of that population, a smaller number of individuals were detected at all three streams in the neighborhood in a given year. Of the entire sample population, 42 individuals were detected in multiple years, 11 of which were detailed in all 3 years. However, none were detected in all 4 years, from 2012 to 2015. 3 individuals were shown to have switched neighborhoods between the years. In 2014, there was 1 individual who switched neighborhoods within the same year. There were no other instances of switching neighborhoods in the same year that were recorded during this study. Non-invasive genetic sampling confirmed a large number of brown bears congregate along Alaskan spawning streams in the summer. Studies have shown much larger numbers of bears forage at waterfalls, such as Brooks and McNeil Falls. However, this study reveals that a significant number of bears are utilizing the spawning streams and acting as vectors between the terrestrial and aquatic environments. The result of the study affirmed the predictions that there would be a frequent movement within foraging neighborhoods, but little movement between them. This is thought to be because bears gravitate towards areas that are predictable in order to increase foraging efficiency. They also return to areas that have a familiar socio-behavioral experience. The results refuted the prediction that there would be a statistical difference between male and female abundance rates. However, females exhibited a greater fidelity in a particular stream within a given neighborhood than males. This is unsurprising because female mammals tend to display this characteristic in comparison with male mammals, who are more likely to disperse. The result of this study showed that the stream neighborhoods for salmon foraging during the summer act as an important and driving force in movement and population along the North Pacific Rim for brown bears.